Many processes, like for instance the expansion of invasive species, can only be understood from a spatial perspective. We will learn in this course how this can be achieved by building on the earlier taught modeling approaches. The models you developed so far were all lacking spatial structure. They thus assume some homogeneous mixing, so processes that are generalized within the considered spatial domain. Such approaches are simple but not realistic. Contaminants, for instance, spread in rivers and form non-uniform gradients. Species invade and leave clear spatial signatures. The dynamics therefore need to be represented in at least one dimension, the dimension of spread. The same holds for biochemical cycles, where sinking of nutrients create variation in trophic interactions along a depth gradient. The processes may here thus even proceed in 3 dimensions. And prey-predator dynamics are by definition heterogeneous in space, because both prey and their predators are mobile and continuously changing locations. Spatial models can be discrete in their spatial representation. Landscapes can, for instance, be represented by discrete cells with distinct properties. Discrete patches, irregular grids representing, for instance, bird territories, or spatial automata where dynamics between grid cells are driven by simple transition rules. As for any model, spatial models differ in their level of complexity, with mathematical models neglecting details of decision-making or heterogeneity on the one hand, and complex individual-based simulation models on the other extreme. We here focus on the most simple and general models, but students in biology will develop their more complex models within some weeks. We have been using so far ordinary differential equations to analyze zero-dimension models. These may, however, still include some spatial aspects, by import and export processes, for instance. The concentrations of nutrients, matter of organisms in a single box, a lake for instance, or something alike, depend for instance on the inflow and outflow processes with dilution going on in the single box. Changes in concentration are the simple sums of flow and dilution rates. These models thus assume a single box, but do not consider spatial processes within this box. We solve this by moving towards 1, 2, or 3 dimensions and the use of partial differential equations, as local processes are now going on along both the time and space dimension. We follow the same reasoning as for the zero-dimension models with in and outflow, but now instead of keeping these processes constant, we make them dependent on the flow processes in the neighboring boxes, which are presented here as cylinders. For each box, at each time interval, the mass of matter in each box of a certain volume delta V is a difference between the fluxes of matter into and out of the box. As for the zero-dimensional model, these depend on the area of the zone of in or outputs times the flux. The mass of the matter in each box is then the volume delta V times the concentration, and changes in the concentration are then the differences in mass in and out divided by the volume, in which the volume can be approximated by the surface halfway between the in and outflow position, x, and delta x plus x, times the surface of the cylinder. And as we are interested in models with continuous space, we shrink the length of the boxes and make them infinitively small. At any point along the cylinder axis, we can thus imagine an infinitively narrow box. Technically, we do that by taking the limit of the above equation for delta x tending to 0. We then obtain a partial differential equation that expresses the rate of change in concentration C as a function of the fluxus J and the surface area A at any point in the domain. 2 types of fluxes can be separated. On the one hand, directional fluxes, which we call advective fluxes. These fluxes then scale with the concentration. And diffusive fluxes, caused by random movements, so-called Brownian motions. These fluxes depend on the concentration gradient and increase when concentrations are larger. Or more visually, imagine a dye spill along one dimension. At t0, the dye is introduced. It is then moved by the current to the right after 20 time steps. The dye is not spread, as it only moved directionally. In case currents were absent, then the dye could diffuse only, so locally spreading out. And of course in reality, both processes will occur jointly. Or for a real illustrative example on dye markers put in sea for drowning men. When released and the man is left behind and few currents are present, the dye is diffusing around the point of release. When the man is still attached to the ship, which is moving, advection is created at the velocity of the ship's speed. And if currents are strong, both processes are occurring simultaneously, even if the man is not attached to the ship anymore. Fluxes for advection are then the advective velocity times the concentration in the box, with length per time as a unit. Examples are net flow through systems, sinking of particles, etc. Diffusion is modeled according to Flick's first law, stating that it depends on the concentration available and the speed of diffusion through space, as determined by the medium, for instance. As diffusion leads to a loss of matter, the sign of the flux is negative. The gradient thus reduces in time. Examples are tidal mixing of nutrients, seed dispersal from trees, etc. So in summary, for a one-dimensional spatial model, the general mass balance in the box is determined by the difference between in- and outflow, plus the local reaction processes that alter the concentration. Consumption, excretion, for instance. Fluxes are the result of convection and advection, and they give rise to the following diffusion, advection, reaction equation. To solve such an equation, we need to define the boundary conditions. If we assume the surface and velocities to be constant, the equation is simplified to three separate terms. And as earlier, we need to define the initial conditions, thus the concentrations in each box at time 0, but also the boundary conditions, as these determine the import and export to the entire box. For advection, upstream concentrations just out of the box need to be defined, while for diffusion, concentrations at the two sides need to be defined for a one-dimensional spread model. These boundary conditions increase in number when spread is modeled along more than one dimension. We can specify boundary conditions in three ways: by imposing fluxes, gradients, and concentrations. For advection, such boundary conditions need to be defined only for the upstream part. The left one defines the concentration in the outside world. The second one, the gradient, which is put to 0, implies there is no diffusive flux. Or boundary conditions of the third kind, on the right, reflecting fluxes outside the considered world. The same holds for diffusion, but now conditions are defined for the two sides of the entirely considered spatial domain. We thus need the partial differential equation and the boundary conditions to find a solution. Again, we may want to find steady state and transient solutions by analytical and numerical approaches. As solving the equations is analytically complex, we rely again on R to do the job for us. We use the react-tran to approximate spatial gradients. We need to define the model grid size, the diffusion and advection parameters, and the different model functions where transport is separated from reaction. So code-wise, we define first the spatial grid, the total length, number of boxes, grid sizes, and mean positions. We define the state variables, parameters and boundary conditions, define the model function, solve the transport and reaction processes, and finally, create the output files by calling the partial differential equation solver, and again, plotting is easy. As we model changes in matter over multiple dimensions, spatial gradients can be plotted at one time step, the right-hand figure, or across time and space with color representing the density, the left figure. This figure shows a clear spread in time with matter accumulating downstream by the end of the simulation, at time step 3000. Now it is up to you.